Well, what is it then that causes the formation of these cytokines? Glial cells, or the brain's macrophages, become activated. And when these glial cells become activated, cytokines are produced. These cytokines then bind to the cell surface and ultimately induce transcription systems which cause the formation via gene activation of these toxic gene products, which then lead to inflammation and pre-programmed cell death or cellular apoptosis. Again, when we're visualizing our patients having a progressive decline, these are the concepts that we have to keep in mind, that essentially the brain is on fire and that we are inducing toxic gene products. So I think it's very important for us to take a very close look and focus on what is it that is activating these glial cells. What activates microglia? Well, one very important concept that certainly relates to dietary interventions is the fact that glucose plays a profound role in this whole scheme. Glucose itself does, in fact, activate microglia. Let's focus on glucose. When we look at glucose and its role, it's important to recognize that glucose can modify proteins in a post-translational way. That is to say that glucose can bind to proteins after they have been manufactured and can bind to the amino group, a free amino group of protein, causing the formation of what we call advanced glycosylation end products. Now, perhaps the most important advanced glycosylation end product that we need to concern ourselves with is beta amyloid that I referred to earlier. Why is this important? Well, when beta amyloid is activated or bonded to glucose, this enhances its production of reactive oxygen species and increases the activation of the microglia. So we need again to focus on this activation of the microglia because that is truly what puts into motion this entire cascade, ultimately leading to gene activation and cellular death. Maureen Reagan was recently in the newspaper, and she indicated a couple of things that I thought were actually quite profound, although they were only mentioned in passing. First, she stated that her father, President Reagan, was a handsome devil with a sweet tooth. And that's important because it sort of helps us understand that sugar and the consumption and the high glucose in the body may in fact predispose or enhance the production of free radicals. Second, the article talked about a study saying that protein deposit may cause Alzheimer's disease. Well, we know now that the protein they are referring to is beta amyloid. And it's the intricate interaction between beta amyloid and glucose that enhances the free radical production. Well, this is certainly borne out by the Rotterdam study of diabetes, recognizing that the relative risk of dementia with diabetes is about 2 times average. The relative risk of Alzheimer's type of dementia is again twice normal, but the relative risk of having dementia if a patient is using insulin is about 400% normal. Now, why would an individual using insulin have such an increased risk? Probably because they've had more events of hypoglycemia, number one, and number two, they've probably had the disease, the diabetes, a lot longer. Now, what else activates microglia? Our list then takes us to the idea of toxins. There's no question that with respect to neurodegenerative diseases, the prototypic toxin is something called MPTP. MPTP is a synthetic form of the drug Demerol, manufactured in the early 1980s. Individuals would shoot this drug intravenously, and it was found that a small percentage of these individuals who had used this drug intravenously suddenly developed fulminant Parkinson's disease. Now, I don't say Parkinsonism because, in fact, these individuals had full-blown Parkinson's disease. We make that statement because, in fact, they were responsive to standard L-DOPA therapy, which is really one of the hallmarks of calling it the disease. These individuals, in addition, had progressive disease, and we'll explore that in just a moment. Well, we know how MPTP works, as you can see by this slide. In the presence of monoamine oxidase type B, MPTP becomes activated to a more toxic intermediate. Now, this toxic intermediate binds specifically to the dopamine transporter, and in so doing does 3 things, as indicated by the 3 red arrows on the right. First, it compromises the mitochondrial's ability to produce ATP, or manufacture energy. Second, it enhances free radical production, or the generation of reactive oxygen species. And third, it activates nitric oxide synthase, the rate-limiting enzyme in the production of nitric oxide. And we will learn later in this series about how important nitric oxide is in the formation of brain free radicals. But what's even more compelling about the MPTP story is that not only did these individuals develop instantly Parkinson's disease, but even long after, the immune response in their brains was persistent. This study demonstrated that post-mortem evaluation of the brains of several MPTP patients showed that, in fact, microglia were surrounding the regions of brain degeneration long after the MPTP had done its damage. Some 12 years later, there is still a neurodegenerative situation which is inflammatory. Other exotoxins are known to predispose to Parkinson's disease, and certainly these have been well described. But I think it's important, especially in our modern society, that we take a few minutes and recognize the profound role that pesticides may have in making this disease more common. And this certainly is not new information. In this study published in 1990, it was found that in Parkinson's disease, there was a 500% increased risk of individuals living in 3 kibbutzim in Israel. Now, kibbutzim are localized regions where individuals share work, they share habitation. But what was found in this study is that these 3 kibbutzim, in which there was such a high incidence of Parkinson's disease, actually shared a water source. And that certainly points a finger to some form of environmental influence. The risk of Parkinson's disease with exposure to pesticides, farming, well water, and rural living was described in 1998. And again, as you can see in looking at this slide, a significant increase risk of Parkinson's disease when individuals are exposed to various herbicides, insecticides, even consumption of well water, a small increased risk. But living on a farm, actually because of these environmental influences, does increase the risk of Parkinson's disease. Dr. Semchuk, writing in Neurology in 1992, stated, quote, "Having a history of occupational herbicide use resulted in a significantly increased Parkinson's disease risk of about 3-fold. Dr. Longstreth found that there was a similar increased risk of amyotrophic lateral sclerosis or Lou Gehrig's disease in individuals exposed to pesticides during their 20s and their 30s. And what's important about this finding was that typically ALS has its onset later in life, and oftentimes it's hard to make the connection between the onset of a disease like ALS and the previous environmental exposure happening maybe 10 or 20 years before. Dieldrin happens to be a very powerful pesticide, and what we know dieldrin does is it's a mitochondrial poison, again affecting the actual energetics of the neuron. It is known that individuals who inherit specific abnormality of a hepatic detoxification enzyme, CYP2D6, which is a P450 enzyme, if individuals inherit a defect of this enzyme, they are at substantially increased risk of Parkinson's disease. We know further, based on Dr. Stevenson's work back in 1989, that if an individual has Parkinson's disease, there's a very good likelihood that that individual has a sulfation abnormality, another important part of phase 2 liver detoxification. As he stated, Parkinson's disease patients may be unusually susceptible to exogenous or even endogenous toxins. And again, that's based upon this flaw of hepatic detoxification in phase 2. Sulfate conjugation, in fact, is very important for the reasons described in this slide. Amine neurotransmitters, of which dopamine class resides, as well as drugs and certainly xenobiotics, these types of detoxification or biotransformation rely heavily upon a sulfate conjugation. In motor neuron disease, in fact, Dr. Stevenson has also found severe abnormalities of sulfation, of deconjugation, of detoxification rather, because of poor sulfur oxidizing and sulfur conjugation activities. These, as he stated, were overrepresented in the motor neuron disease group, and in fact, Dr. Stevenson has also demonstrated similar findings in patients suffering from Alzheimer's disease. So what he has said is that individuals suffering from Lou Gehrig's disease, or ALS, Parkinson's disease, and Alzheimer's disease all have deficiencies of a very important pathway of phase 2